A very good morning, dear students. Today we will see the production of bile pigments. Bile pigments are normally produced from the degradation of heme. Last class we have seen the production of heme, and today we will see the production of bile pigments, that is by the degradation of heme. Normally the erythrocytes, red blood cells, are having a lifespan of 120 days. After this 120 days, it is taken up by the macrophages, that is degraded by the macrophages of the reticuloendothelial system in spleen and liver. The hemoglobin is degraded into heme and globin. Globin is the protein, and heme we have seen the non-protein part of hemoglobin. Globin again goes into the production of hemoglobin or degraded into its constituent amino acids. Okay, hemoglobin degraded into heme and globin, and here globin goes back to the production of hemoglobin or it is degraded into its constituent amino acids. Then we have to study the degradation of heme. What happens to the heme? Heme, by the action of an enzyme heme oxygenase with the help of coenzyme NADPH and oxygen. With the help of NADPH, coenzyme NADPH and oxygen, and with the help of enzyme heme oxygenase, heme is degraded into a compound known as biliverdin. Biliverdin is a green color pigment, and here we have to see one thing. See, this is a porphyrin ring, and here the methenial bridge of A and B pyrrole ring. A, B, C, D. There are 4 pyrrole rings, and here the A methenial bridge between the pyrrole ring A and B is broken and forms this biliverdin. And here also produces a molecule of carbon monoxide. Then this carbon monoxide is the only production of endogenous carbon monoxide. Here is the only production of endogenous, that is inside the body, endogenous production of carbon monoxide, and ferrous ion in the heme is expelled as ferric ions. So heme is degraded into biliverdin by converting or cleaving methenial bridge between A and B pyrrole ring and forms a green color pigment known as biliverdin. Heme നമ്മുടെ എന്താ പറയുന്നത് ഹീം ഓക്സിജനൈസ് എൻസൈമിൻ്റെ സഹായത്തോടെ അല്ലെങ്കിൽ കോഎൻസൈം NADPH ഓക്സിജൻ്റെ സഹായത്തോടുകൂടി പൈറോൾ A ആൻഡ് B പൈറോൾ റിങ്ങിനിടയിലുള്ള മെത്തീനിയൽ ബ്രിഡ്ജ് ബ്രേക്കായി ഒരു ലീനിയർ കോമ്പൗണ്ട്, പൈറോൾ റിംഗ് 4 ലീനിയർ ആയിട്ടിരിക്കുന്ന കോമ്പൗണ്ടായിട്ട് കൺവേർട്ട് ചെയ്യും. ഇതിൽ Fe2+ ഫെറസ് അയോൺ ഫെറിക് അയോണായിട്ട് എക്സ്പെൽ ആവും, ഒരു മോളിക്യൂൾ കാർബൺ മോണോക്സൈഡും പ്രൊഡ്യൂസ് ആവും, ബിലിവെർഡിനായിട്ട് കൺവേർട്ട് ചെയ്യും. ബിലിവെർഡിൻ ഒരു ഗ്രീൻ കളർ പിഗ്മെൻ്റ് ആണ്. ഈ ബിലിവെർഡിൻ എന്ന് പറയുന്നത് ബേർഡ്സിലും ആംഫീബിയൻസിനും എല്ലാം ഈ ബിലിവെർഡിൻ ആസച്ചാണ് എക്സ്പെൽ ആവുന്നത്. പക്ഷേ ഹ്യൂമൻസിന് അങ്ങനെയല്ല. മാമൽസിന് biliverdin is further converted into bilirubin with the help of enzyme biliverdin reductase. with the help of enzyme biliverdin reductase, biliverdin is converted into bilirubin. Bilirubin is a yellow color pigment. Here also coenzyme NADPH is required. And here the methenial bridge CH, methenial bridge of C and D ring is converted into methylene ring. This methenial bridge is converted into methylene bridge between this C and D. That is a change in converting biliverdin into bilirubin. അതായത് സി പൈറോൾ റിങ്ങിനും ഡി പൈറോൾ റിങ്ങിനും ഇടയിലുള്ള മെത്തീനിയൽ ബ്രിഡ്ജ് ഡബിൾ ബോണ്ട് സി എച്ച് സിംഗിൾ ബോണ്ട് എന്നുള്ളത് മാറി സിംഗിൾ ബോണ്ട് സി എച്ച് ടു സിംഗിൾ ബോണ്ട് ഇങ്ങനെ മെത്തിലിൻ ബ്രിഡ്ജായിട്ട് കൺവേർട്ട് ചെയ്യും. ഇതാണ് ബിലിവെർഡിൻ റിഡക്റ്റേസ് ചെയ്യുന്ന ചേഞ്ച്. സോ ദിസ് ഈസ് എ പ്രൊഡക്ഷൻ ഓഫ് ബിലിറൂബിൻ. സോ hemoglobin degraded into heme and globin, and heme further degraded into a compound biliverdin with the help of enzyme heme oxygenase, and it is further converted into bilirubin, an yellow color pigment, with the help of enzyme biliverdin reductase. This is the formation of bilirubin. And now we have to see the transport of bilirubin. Transport of bilirubin. This bilirubin combines with albumin. Bilirubin is a lipophilic compound. Bilirubin is a lipophilic, that is non-soluble in aqueous solutions. So it is lipophilic. It combines with albumin and transported to the liver. Transported to the liver. And in liver, what happens here? This is a sinusoidal cells. This is the consider this is hepatocyte. Here it enters into the sinusoidal space and cleaves into albumin and bilirubin. See, after entering into the sinusoidal space, this complex, albumin bilirubin complex, is degraded into albumin and bilirubin, and this bilirubin enters into hepatocytes with the help of carrier mediated diffusion. And inside the hepatocytes, this bilirubin combines with a protein Y and another protein known as ligandin. See, inside the hepatocyte, this bilirubin combines with a protein known as ligandin and protein Y. And this combination, this formation of this complex prevents the backflow of bilirubin into the system. അതായത് ബിലിറൂബിൻ സൈനുസോയിഡൽ സ്പേസിൽ കയറിയ ഉടനെ ആൽബുമിനും ബിലിറൂബിനുമായിട്ട് സ്പ്ലിറ്റ് ആവും. സ്പ്ലിറ്റ് ആയ ശേഷം ഒരു കരിയർ മീഡിയേറ്റഡ് ഡിഫ്യൂഷനിലൂടെ ഇത് ഹെപ്പാറ്റോസൈറ്റിനുള്ളിലേക്ക് വരും. ഉള്ളിൽ വന്നു കഴിഞ്ഞാൽ ഇത് ബിലിറൂബിൻ രണ്ട് പ്രോട്ടീനുമായിട്ട് കമ്പൈൻ ആവും, ലിഗാൻഡിനും പ്രോട്ടീൻ വൈയും. ഇത് ബിലിറൂബിൻ്റെ ബാക്ക് ഫ്ലോ ഹെപ്പാറ്റോസൈറ്റിൽ നിന്ന് പുറത്തേക്ക് പോകുന്നത് പ്രിവെൻറ്റ് ചെയ്യും. മാത്രമല്ല ഇറ്റ് ഹെൽപ്സ് ടു കീപ്പ് ഇൻ സൊലുബിലൈസ്ഡ് ഫോം ഓഫ് ബിലിറൂബിൻ. ബിലിറൂബിനെ ഒരു സൊലുബിലൈസ്ഡ് ഫോമിലേക്ക് കൺവേർട്ട് ചെയ്യാൻ, അതായത് കോൺജുഗേഷന് വേണ്ടി സൊലുബിലൈസ്ഡ് ഫോമിലേക്ക് മാറ്റാൻ വേണ്ടി ഹെൽപ്പ് ചെയ്യും. ഓക്കെ, അപ്പം ഇവിടെ നമ്മൾ അറിയേണ്ടത് ഇതൊരു റേറ്റ് ലിമിറ്റിംഗ് സ്റ്റെപ്പ് അല്ല, നോട്ട് റേറ്റ് ലിമിറ്റിംഗ്. എത്ര ബിലിറൂബിൻ ആൽബുമിൻ കോംപ്ലക്സ് വന്നാലും ഹെപ്പാറ്റോസൈറ്റിലോട്ട് കയറിക്കോളും. ഓക്കെ, അപ്പോൾ ഇതൊരു റേറ്റ് ലിമിറ്റിംഗ് സ്റ്റെപ്പ് അല്ല. ഓക്കെ, അതൊന്ന് ഓർമ്മിച്ച് വെച്ചോളണം. So bilirubin combines with albumin and transported to the liver. Inside the liver, the complex enters into sinusoidal space and splits into albumin and bilirubin. And through a carrier-mediated diffusion, bilirubin enters into hepatocytes and combines with proteins known as ligandin and protein Y. This prevents the backflow of bilirubin and it helps to keep the bilirubin in solubilized form before conjugation. Okay. അപ്പോൾ ഇത്രയും കാര്യങ്ങളൊന്ന് ഓർമ്മിച്ച് വെച്ചേക്കണം. നെക്സ്റ്റ് ഈസ് കോൺജുഗേഷൻ. Conjugation of bilirubin. Bilirubin combines with glucuronide with the help of bilirubin glucuronyl transferase enzyme. Bilirubin glucuronyl transferase enzyme. Bilirubin accepts a glucuronide molecule from UDP glucuronide. Bilirubin accepts a glucuronide from UDP glucuronide with the help of bilirubin glucuronyl transferase enzyme, and it forms bilirubin monoglucuronate. And then again with the help of same enzyme bilirubin glucuronyl transferase, it accepts one more molecule of glucuronide from UDP glucuronide, forms bilirubin diglucuronate. So this is a conjugation of bilirubin. By this conjugation, lipophilic bilirubin is converted into hydrophilic bilirubin. This is soluble in aqueous form. So bilirubin accepts a molecule of glucuronide from UDP glucose with the help of enzyme bilirubin glucuronyl transferase, and it forms bilirubin monoglucuronide. And again with the help of the same enzyme bilirubin glucuronyl transferase, and it accepts another molecule of glucuronide from UDP glucuronide, forms bilirubin diglucuronide. It is a soluble form of bilirubin. That is conjugated form of bilirubin. Okay. And a small portion of this bilirubin, this small portion of bilirubin is enters into enterohepatic circulation again, and major portion of this bilirubin diglucuronide conjugated form is excreted into the bile or through bile canaliculi. Major portion of this conjugated bilirubin is excreted to bile through bile canaliculi, and a small portion is re-enters to the enterohepatic circulation with the help of protein multi-drug resistance protein. Multi-drug resistance protein 3. A small portion of this conjugated bilirubin enters into hepatic or portal circulation with the help of enzyme, with the help of protein multi-drug resistance protein 3. And this conjugated bilirubin is reabsorbed into the portal or liver by the or hepatocytes by the proteins known as organic anion transporter 1b1 protein. Transporter protein 1b1, that is OAT1b1, and organic anion transporter protein 1b3. So a major portion of conjugated bilirubin is excreted through bile, and a minor portion is enters into portal circulation with the help of protein known as multi-drug resistance protein 3. And this conjugated bilirubin which enters into the portal circulation is reabsorbed by the hepatocytes or liver by two proteins known as organic anion transporter protein 1b1 and organic anion transporter 1b3. And a defect in this or modification of this protein, a defect in these two proteins leads to a syndrome known as Rotor syndrome. Okay. Then while we have told this bilirubin diglucuronide or the conjugated form of bilirubin is excreted into the bile canaliculi. This is against the concentration gradient. Concentration gradient. The excretion of conjugated bilirubin into the bile is against concentration gradient, or it is an active transport, and this is the rate limiting step. A rate limiting step. We have to remember the specific point. The rate limiting step in bilirubin metabolism is the excretion of conjugated bilirubin into the bile. Okay, that is a rate limiting step, and this active transport is with the help of protein known as multi-drug resistance protein 2. The excretion of conjugated bilirubin into bile is with the help of multi-drug resistance protein 2, and a defect in this multi-drug resistance protein 2 leads to a disease known as Dubin-Johnson syndrome. A defect in multi-drug resistance protein 2 leads to a syndrome known as Dubin-Johnson syndrome. Again, next is the fate of bilirubin metabolism. Bilirubin from the bile enters into the intestine. A major portion of bilirubin with the help of enzyme beta glucuronidase, enzyme produced by bacteria. A major portion of conjugated bilirubin. A major portion of bilirubin in intestine is converted into bilirubin, unconjugated form, unconjugated bilirubin, and almost 90% of this unconjugated bilirubin is converted into stercobilinogen or stercobilin. This is excreted through feces, and this stercobilinogen produces a brown color to the feces, and approximately 10% of this unconjugated bilirubin is excreted by kidney, and it is converted into urobilin. It's an yellow color pigment which gives yellow color to the urine. Approximately 10% of unconjugated bilirubin is converted into urobilin and excreted through urine. That gives this urobilin gives yellow color to urine. One gram of hemoglobin normally gives 35 milligram of 1 gram of hemoglobin produces 35 milligram of bilirubin. 1 gram of hemoglobin approximately produces 35 milligram of bilirubin. Daily approximately 6 gram of hemoglobin is degraded and produced by the body. Daily approximately 6 gram of hemoglobin is produced as well as degraded by body. Okay, this is all about the metabolism of bilirubin. Okay, thank you.